let me start the recording. And now let me... Okay, so once I click over, somebody with a microphone, tell me if you can see this, just so I don't have to click back and forth.
Yes.
All right, thank you. All right, so test 2 survival guide. So the exam is going to be set up exactly like last exam. Friday, your individual exam will open at 5 o'clock, and it will close Sunday at 11:59, so you'll have the whole weekend to take it. You'll have 40 minutes to take the exam. It's about 30 questions, and then, of course, the bonus questions, which is separate, so make sure that you take both. And then next Monday, so a week from today, you will do the group exam exactly like we did last time. Okay? All right. So let's get into the meat and potatoes of it. So, law of thermodynamics. You have the first law of thermodynamics, which is basically just saying that energy cannot be created or destroyed, only transferred or transformed. So this is like, if I am out of energy, if I am exhausted, I can't just poof out some energy. I have to eat some food or take a nap or drink some coffee or eat something sugary. I have to do something to get some energy. I can't just snap my fingers and be the Energizer Bunny. Wouldn't it be really cool if it worked that way? So an application to the first law of thermodynamics is just saying that an organism must obtain their energy from the environment. So this is like if a bunny eats the grass is how the bunny gets its energy. Now say, I don't know, a fox. Do fox eat bunnies? We'll say they do. Let's say a fox eats the bunny. Then the fox is going to get 100%, well, not 100%, but a fox is going to get its energy from the bunny. And then it's just up the food chain like that. Okay? Now, the second law of thermodynamics just says that every exchange of energy increases the entropy in the universe. So, basically, what entropy is just a disorder. It's saying that in every exchange of energy, there are imperfections. So let's say a fox eats a bunny. 100% of the bunny's energy is not going to that fox. Some of it will be lost in heat, and the heat just kind of goes out into the environment. So entropy is just saying that nothing's perfect. That no matter how you do it, there is some messiness to it. Some of the energy is lost. Okay, so now moving on to catabolic and anabolic. Guys, I cannot stress this enough. You need to be able to do this whole chart. This whole chart. How I would study is give yourself one. So either yourself the title or say like break down complex molecules and try to fill everything else out. Okay? I cannot stress this enough, okay? This is the best way I have found to learn it. It's a good way to test yourself. And if you remember, like in a chart form, it's easier to connect them on the exam. Okay? Okay, so a catabolic reaction is exergonic. It has a negative free energy. It's spontaneous. It breaks down complex molecules. It releases energy. The reactants are higher than the products. And an example is hydrolysis. So let's break this down. This is actually wrong. It's technically a catabolic pathway. So let me change that. It's a catabolic pathway. And then the reaction type is exergonic. Remember, the energy is exiting. So this is the one where a graph looks like a slide. So remember, when you make a graph, you always start with the products and then you end with the reactant. Sorry. Whoops. flip that. You always start with the reactants and you end with the products. I am so sorry, guys. It's been a Monday. So you start with the reactants and you end with the products. So if the reactants are higher than the products, then it's a slide or a declining graph. And that is how you get the negative free energy. So think of this like a negative slope is how I like to think of it. All right, so also when it comes to spontaneity, I like to think of this like you're standing in the middle of a flight of stairs. I can spontaneously fall down the stairs. I'm probably not going to try to do it. I'm definitely not going to think about it. I'm not going to put any energy into falling down the stairs. It's just going to happen. And I'm probably going to yell when I'm falling down the stairs. And that's releasing some energy. So I didn't put anything in. I just spontaneously fell down and I'm yelling on the way down. That is like a catabolic pathway. Okay? So a catabolic pathway is used to break down complex molecules. And an example of this is hydrolysis. So I know, guys, but this stuff does not go away. So back in our biomolecule section, we talked about how if you have a polymer and you want to make two monomers, you have to do hydrolysis, which is just adding water to break one thing into two. That is technically a catabolic pathway. Okay? All right, so let's go to the counterpart, which is an anabolic pathway. Okay, so it is an endergonic reaction. So endergonic means that the energy needs to enter. It consumes energy, so you need to have some energy to even start it. So let's go back to our stair example. I'm standing in the middle of a flight of stairs. If I want to climb the stairs, if I want to go up, then I have to think about it, I have to tell my feet to move, and I have to walk. I have to think about it. I have to put some effort or some energy into doing it. I can't just spontaneously levitate up the stairs. I have to make my muscles move and walk up the stairs. That means it's non-spontaneous. I can't spontaneously do it. And also because you're going up, the slope of the overall graph is positive. So it's got a positive delta G. And because it is an increasing graph, the reactants are lower than the products. So you end higher than you start. Okay? And back to our biomolecule section, if I had two monomers and I wanted to make them one polymer, I would use a process known as dehydration synthesis, which is an anabolic pathway because you build a complex molecule. Okay? Okay, so I need somebody to, so you can either unmute yourself or you can put in the chat. Either is fine. So what kind of graph is this? Is this catabolic or anabolic? Good job, guys. Good, good, good. I love it. So this graph is catabolic. So here's your reactants. Here's your products. The overall thing is decreasing. All right. Well, I was going to ask you what this hump was, but it says it right here. So the hump is the activation energy. This is what is required for the whole process to start. So this is like our speed bump. Remember last time driving on campus, but you can't go over 15 miles an hour, so you gotta go over the speed bump, and you're going kind of slow, and then you get off of campus, and you're on McCorkle Avenue, and you go shoop, and then you get home, so you're slowing down. Okay? Now, the orange line represents this without a catalyst or an enzyme, and the dotted line is with an enzyme. So this is kind of foreshadowing, because I will talk about this more in depth, but the main thing of this is that with an enzyme, you lower activation energy, and therefore you increase the rate of reaction or the speed of the reaction. Okay? Okay, feedback inhibition. So this is a pathway where the end product will shut down the pathway itself. So I like to think of this like, say you have a bunch of third graders line up and you're going to take them to lunch. This is back when we actually had in-seat school. So you line up the third graders and you're going to take them to lunch, and you would say, last one out, shut the door and turn off the lights. So the last one out, the last one that is needed, or the last one that is there, is going to shut everything off and go about its business. But when, say, the third graders are back from lunch, the classroom is still there, the light switch still works, you're not locked out, it's still there, just the last product turns it off. So the main purpose of this is to prevent waste, so that the cells don't make more than they're needed. And this is her cupcake example, so don't make too many cupcakes. So her example in class was you can only make enough cupcakes to fit the counter space that you need. Excuse me. So you can only make as many cupcakes as you have space. Okay? All right, moving on to enzymes. So an enzyme is specific reaction, So enzymes have a specific reaction that they catalyze. An enzyme can't just catalyze any reaction, they're specific. The substrate is the reactant of a reaction. It's the reactant that binds to an enzyme. The enzyme substrate complex is actually when the substrate is bound to the enzyme. The active site is where the substrate binds to the enzyme. Okay, so who can tell me what an enzyme is? It's our favorite biomolecule. Protein, exactly. An enzyme is a protein. Okay, so the whole purpose of an enzyme is that it speeds up or increases the reaction rate. So that's the main thing that it does. And it does this by lowering the activation energy. So instead of the speed bump on campus being 6 inches thick, say an enzyme is going to cut it down to 3 inches thick. So instead of having to go 15 miles an hour on campus, you can go 30 miles an hour on campus. You still have to cross that speed bump. There is still a toll to pay. There is still something regulating, but it's just not as high as if the enzyme wasn't there. Okay, does all of this make sense? Because if so, I'm on to cellular respiration, which I know a lot of you are struggling with. So let me click back here. Are there any questions about catabolic versus anabolic or enzymes or anything of this nature? Okay. All right, so let's switch over to cellular respiration. So, I would say knowing the end products of every step is going to be extremely, extremely, extremely important. So make yourself a chart. I know I sound like a broken record, but the best way to study for this exam in a lot of ways is to make charts. In glycolysis, this is what you put in, this is what you get out. In the prep step, this is what you put in, this is what you get out. And make sure to do that for everything, okay? That's the best way to study. For glycolysis, you do not require any oxygen, so it can be aerobic or anaerobic. So who wants to tell me what anaerobic means?
It means it doesn't require oxygen.
Exactly. So then what would aerobic mean?
Requires oxygen.
Exactly. So in glycolysis, if oxygen is there, cool. If oxygen is not there, it doesn't matter. It's not going to use it either way. It's almost like inviting a vegetarian friend over. If you have ground beef in your freezer, it doesn't matter because they're not going to use it. If it's there, cool. If it's not, cool. They're indifferent to it. Okay? So the starting thing, the starting molecule is a glucose, which is 6 carbons. So when you're making water in, say, the citric acid cycle or something of that sort, the oxygen comes from the glucose itself. So molecular oxygen, the oxygen that you get with ventilation, does not come in until oxidative phosphorylation, the electron transport chain, and all of that stuff. So right now, the only oxygen that we have is from the glucose itself. Okay, so to start glycolysis, you have to pay a toll, and it requires 2 ATP. It occurs in the cytoplasm. All right, so now we are having with the products. So it produces 2 net ATP. So remember, you started with 2. You get 4 overall, but only actually gain 2. So the only thing she'll really ask you about is the net, how much you actually gained. It also produces 2 NADHs, which are the electron carriers, and you end with 2 pyruvates, which are 3 carbon molecules. Okay? Now moving on to the prep step. Excuse me, y'all. So now we're going to move on to the prep step, which another name for that is pyruvate oxidation. So, hold on. Let me back up real quick. So you should be able to, for every step, you should be able to answer, does it require oxygen? Where does it occur? What do you start with? What do you end with? That is the best way to test yourself. Glycolysis, what do you start with? What do you end with? Where does it occur? Make a chart, make flashcards, just make sure you know that for every step, okay? Okay, so the prep step does not require oxygen. It occurs in the mitochondria, and you produce 2 Acetyl CoA, and you produce one of the NADHs, which is an electron carrier. Okay, so now moving on to the citric acid cycle. This is also called the Krebs cycle, but I believe she refers to it as the citric acid cycle on your exam, but just know your book or other sources may also call this the Krebs cycle. So your starting product with the citric acid cycle is 2 Acetyl CoA and 2 Oxaloacetate. So that's why the citric acid cycle is formed in a circle. You start with Acetyl CoA, turn that into Oxaloacetate, and then you're just back in a huge circle to end with Oxaloacetate. Okay? This is just a big circle. And she doesn't really care the reactions that happen in that circle. You just need to know that it occurs in the mitochondria. It does require oxygen. So this is your first step that you cannot do without oxygen. So that makes it aerobic. And you produce 2 of the FADH2, 6 of the NADH and 2 ATP overall. So this is in 2 cycles. Now, who can tell me why we have to do 2 cycles of the citric acid cycle? Why do we have to do 2?
We start with 2 Acetyl CoA.
Exactly. So you start with the glucose, and then you do glycolysis, which breaks it into pyruvates, and then those are turned to acetyl CoA. So you have 2 acetyl CoAs. That's exactly right. You have 2 acetyl CoA, so you have to go through that twice. That is exactly right. Good job. So in 1 cycle, you would produce 1 of the FADH2, 3 of the NADH, and 1 ATP. Okay? Does that make sense? Okay. Now we're going to move on to the electron transport chain. So the main thing of the electron transport chain is that it sets up the proton gradient. So that picture that she drew in class, that's the main thing that happens in the electron transport chain. It really doesn't produce anything. It occurs in the mitochondria. It does require oxygen. That stays the same. But it doesn't really produce something. It just sets up the proton gradient. So it sets up the proton gradient by moving the protons up their concentration gradient using an electron. So think of this like a smuggler. If you're smuggling illegal chocolate, I don't know. If you're smuggling some kind of special chocolate or something, okay, perfect example. You don't want to pay the movie theater candy prices because who wants to pay that? So you smuggle in a pack of M&Ms and a box of popcorn. You must have a really big purse, but we're going to go with it, okay? So you pay your toll into the movie. You are there legally. You're there rightfully. Your popcorn in your purse is not. It just kind of sneaks its way in. Okay? So that's how this proton gradient works. The protons don't pay. They don't pay. The electrons move up the gradient. So the protons are just along for the ride. Does that make sense? Because I know a lot of you were confused about this today. Okay, so once the protons are up the gradient, the electrons are going to mix with oxygen and 2 hydrogens to make water, which is why when you breathe out, say like if you breathe onto your glasses, like to clean them, that's why they get fogged up, because your breath has water in it. So the electron is going to pair with an oxygen and 2 hydrogen to make a water molecule. Okay? Okay, does this help? Because my class looked very, very confused today, and that's completely understandable because this stuff is very hard. Excuse me.
The picture that she drew today was really confusing.
I got a lot of that, so it's just talking it out. Is that better?
Yeah.
Okay, good.
Also,
Okay.
Prep step, do you have to have oxygen for that or no?
For what step?
For the prep step?
No, you do not have to have oxygen for the prep step.
Okay, thank you.
No problem. Okay, so the last thing about electrons that I want to mention really quick is that electrons are moving through this whole chain. Through this whole cellular respiration cycle, electrons are moving. Through the electron carriers, through the FADH2, through the NADH, electrons are constantly moving. But I want you guys to remember that ATP has nothing to do with the electron pathway. ATP only has to do with creating energy. It has nothing to do with electrons. Electrons is a totally secondary process, and electrons in this case are just used to pump hydrogens up the gradient, and in which case will create ATP. Okay? Okay, so chemiosmosis. This is the second part of oxidative phosphorylation. I will never be able to say that word. It's fine. It does require oxygen. It occurs in the mitochondria. So you start with the proton gradient, electrons, and oxygen. Okay? Okay, so electrons are going to pump down the gradient, which create ATP. Sorry. Protons are going to pump down the gradient. So sorry. Protons are going to pump down the gradient, which creates ATP. The big thing about this step is that the big part of ATP is being created. So this is the last step. This is where you get the kit and caboodle, okay? It produces around 30 ATP molecules per one glucose molecule. So let's say I had 3 glucose molecules. How many ATP would I get? $90, exactly. And you would get to acetyl CoA. That's right. But if we want to go all the way through cellular respiration, you would get about 90 ATP. Okay, so that is all I have for aerobic cellular respiration. Any questions on that? And I'm going to briefly talk about anaerobic because I'm not sure how far she's going to get into it because we are a little bit behind. But cellular respiration is the big thing that I need you guys to be up on, okay? So how do you feel about it? Good, bad, in between? Good, way better now. Okay, good. Good. Okay. All right, so anaerobic respiration just means that it happens without oxygen. So, quiz. How far into cellular respiration can you get without oxygen? So remember, what is the last step that doesn't require oxygen?
The prep cycle.
That's exactly right. It's a prep step. But the thing about the prep step is that it doesn't produce anything. The whole purpose of the prep step is just to move something, to move it from the cytoplasm into the mitochondria. So it doesn't produce basically anything. So the main ATP production is going to come from glycolysis, which is only 2 ATP per glucose molecule. So it's not a lot at all compared to the 30 that we were getting in cellular respiration. So the only ATP producing step is glycolysis. Okay? That's all I'm going to talk about anaerobic because I'm not sure how far she's going to get into it. Okay, now I'm going to talk about mitosis, and I know we have not covered it. I'm not even sure how far she's going to get, but as far as she gets on the end of class on Friday, that's what will be on the exam, okay? Okay, so I'm going to talk about how DNA is packaged. So, DNA can come in 3 packages or types. It can be chromatin, which is kind of like ramen noodles, like cooked ramen noodles. They're all squirmy and they're like all over the place. They're not neat. They're just chilling in a bowl, okay? So this is unpackaged DNA. All right. So sticking with our ramen noodle example, if you take ramen noodles out of the package before you cook them, that whole block is a chromosome. It's packaged. It's neat. It's in a little block. Okay? Now, a sister chromatid is one side of a chromosome. So you all know how ramen noodles have like a split in the middle, and if you like short noodles, you can break it at that split. You all know what I'm talking about? We're all college kids. I know we have eaten many a ramen noodle. Right? So if you split the ramen noodles in half, one side of that is a sister chromatid. Okay? While they're still together. And if this doesn't make sense, that's okay. We haven't gone over in class. I just want to kind of introduce you all to all of this, okay? Okay, so all of this stuff she's going to go over more in detail in class, so I'm going to let her do that. So for interphase, this is when cells aren't doing anything. They're just kind of chilling. They're living their life. So when a cell is in interphase, the DNA is packaged as chromatin, which means that it is unpackaged. It's ramen noodles. They're all loosey-goosey all over the place, okay? They're just kind of living their best life and they're doing stuff. Okay? Okay, so moving on to G1. So there are 3 phases of interphase. G1 is just growth to normal size and you do regular function. So I like to think of these stages as like the stages of life, like human life. So G1 is your whole childhood. Your one job is to grow. You're just growing and there's not a care in the world and there's no stress and you're just chilling and you just have regular functions. The S phase is where DNA synthesis occurs and you duplicate your DNA material, your chromatin. And this is like the college phase, the young adult phase, 18 to 22 phase. You are doing something. You're trying to make something, but you don't really know what you're making. You don't know what your purpose is yet. You're confused all the time. You're stressed out all the time, but you're still doing something. It's just not, you're not seeing the results of that yet. Now, the G2 phase, think about this like you're growing a family. Okay, so you could think about this like a pregnant woman or something like that. You grow to double your normal size. So when someone is pregnant, they obviously grow because they have a human inside of them. So you grow to double your normal size and you produce structures needed in cell division, such as spindles and microtubules. So you're getting ready for the overall division. Okay? All right, so now we're into the mitotic phases. So the mitotic phases are when the cell is actually dividing. So the purpose of mitosis is to make a daughter cell that is an exact replica, a photocopy of the starting cell. So this is different from meiosis, which will not be on this exam, but I don't want, if you've heard that term before, I don't want you to get that confused. Mitosis is when you want to get a daughter cell that is exactly carbon copy like the original cell. So say if I cut my arm, I don't want liver cells to grow like over the cut. I want the same cells that were there. I want the epithelial cells. Okay? So the first step of mitosis is prophase. So this is just where the chromosomes condense. So in prophase is where you go from chromatin to chromosomes. So the centrioles are like the puppet sticks. I think that's what they're called. Like if you have a marionette puppet and you have sticks that you use to control the puppet, that's like what the centrioles are. And they develop spindles that are like the strings. So the spindles and the centrioles are just moving to opposite sides of the cell. That's all that's happening. The chromatid turns to chromosome, and the centrioles and spindles develop. So in prometaphase, the nuclear envelope completely breaks down. So remember, the nuclear envelope is that membrane around the nucleus. The chromosome attaches to spindle fiber. So you're attaching the string to your puppet. And the centrosomes are now on opposing poles of the cell. So the centrosomes are on either side of the cell. The chromosomes are developed. The spindles are attached to the centrosome and to the chromosome. And that's it. They're just attached. Nothing's moving. It's just attached. Okay? Now, metaphase is just where the chromosomes align on the metaphyseal plate, or they meet in the middle. So chromosomes will line up real nice and neat right in the middle of the cell, so that they can eventually be pulled apart real nicely. But metaphase is just them lining up. So they're lining up real nice in a row. So anaphase is the stage of actually pulling them apart. So this is where you're using your puppet sticks to actually move your puppet. The centrosomes are moving the chromosomes apart. So when two sister chromatids are separated, they are now chromosomes. So when you have two together, you can see me. Hold on, let me turn my camera on. Maybe if this will come up. I'm sorry, y'all. Okay, well, it's not going to work right now. I'll try it again at the end, but let's just keep on rolling. So when two chromatids separate, each individual one is called chromosomes. And the chromosomes are pulled apart, and they move to opposing sides of the cell. Now remember, they're pulled apart by those puppet sticks, by the centrioles. Okay? All right, so telophase is the end stage. This is cytokinesis. This is where the nuclear envelope reforms. The cells will cleave because they have two exact pieces of DNA. So it's where the cleavage line forms and they become two separate cells. My camera's working now? Oh, yay. Okay. So what I was going to talk about is, this is when you have sister chromatids. They're separated. So if you're talking about this one, then it's a chromatid. If you're talking about this one, it's a chromatid. If you're talking about the whole thing, so both of these fingers, that's a chromosome. Now, in anaphase, these are going to line up on the plate. So it's going to line up on the metaphyseal plate. Sorry, that's metaphase. In metaphase, they're going to line up in the metaphyseal plate. Think metaphyseal middle. They're going to meet in the middle. Okay? Now, in anaphase, they're going to be pulled apart. So they're two separate now. Now, since they're separate, nothing's changed, but they're separate. They are now known as chromosomes. They're only sister chromatids when they're together. When they're pulled apart in anaphase, they're known as daughter chromosomes, or just chromosomes. Okay? Does that make sense? I know you guys haven't covered it, so it's completely understandable if you're still confused, but I just wanted to kind of introduce it. Okay, now in telophase, each cell has a chromosome. So what happens is, so this is your big cell in mitosis. And then it kind of cleaves in and cleaves in and cleaves in until it's separate like that. Does that make sense? Could you all see that? Okay. All right, I'm going to assume so. My Wi-Fi has been really, really bad lately, so I haven't been able to use my camera very much. Okay, so here I have a photo. I'm going to stop sharing my camera. Oh, am I gone? Can you guys still hear me? Yeah. Okay, guys, I'm going to refresh my page because it froze on my end. So I'm going to refresh and hopefully we'll get back. Okay, guys, I have returned. Nothing is frozen anymore, so we're all good. So sorry about that. So let me share my screen one more time, and we're just going to look at these photos really quick, and then I'll open the floor for questions, and if not, I'll ask you guys questions if you want to stay for like a little quiz, like review thing. Okay. So these are just the stages of mitosis. So if you can see on interphase, you can't really see the chromosomes. All you can see are the nuclei. So the dark staining part is the nuclei. In prophase, you can see the chromosomes. They're condensed. They're just not like lined up. They're just kind of there. In metaphase, you can see they're lined up real nice in a row. In anaphase, they're being pulled apart. So you have chromosomes on either side of the cell. And in telophase, you can see there's chromosomes here, and you can even see a little cleavage line right there. Okay, so that's all I have. I know that you guys have not gone over mitosis. I'm not even sure how much of that will be on the exam, but on the version I saw, there was a few questions on mitosis. Just know that if we don't cover it, it's not going to be on this exam, guys, okay? So don't stress about like teaching yourself mitosis or something. If you don't cover it, it won't be on the exam. If something changes, I'll email you guys and let you know. So be confident, y'all. You got it. I'm really proud of each and every one of you. You all are doing amazing this semester. We are almost done with the semester. We are in midterm season. Well, we're past midterm season. So we got this. All right. So do any of you all have questions? Yes. So say if we only get to metaphase, she could still ask questions about prophase. So wherever we get to on Friday will be the cutoff date. Sure. Go right ahead, Ryan. Yeah. Yeah. So after every study session, I will email you guys out the PowerPoint. That reminds me, I'm going to take a picture of the participants so I know who all was here, because I've been having the memory of a goldfish lately. Okay. All right. So, any more questions? These are really good questions. Anything that you guys are kind of asked about, or that you want to go over, or do you want me to ask you all review questions, or do you just want to leave and go to bed? You all talk to me. This is your session. What do you want to do?
Can you like ask us some questions? I think that would definitely help because I'm definitely confused with the mitosis stuff, and like I'm worried because we only have like so little time to study for that. She puts
Okay.
it on the test.
All right. So, do you want me to ask questions about all of it, or do you want to just cover mitosis?
All of it.
All of it. Okay. So, who can tell me what entropy is? What is entropy? And yes, Eric, I will add Ethan Gundy to the list. I saw that he left mid-session, so I will add him. Chaos, exactly right. It's the level of disorder, the level of flaws. Exactly. So what does that look like in an energy transaction? Those are all perfectly right answers, but what does it look like? Who can give me an example? Heat, exactly. And I love that you put where it's going. So anytime that we say heat is released, it's going to the atmosphere, out into the environment. That's completely correct. Now, which law of thermodynamics is entropy connected to? Okay, so entropy is actually connected to the second law. Yep. So what is the first law? Who can tell me what the first law of thermodynamics is?
Energy cannot be created nor destroyed.
Exactly right. All right, so now let's go into catabolic versus anabolic. So if the only thing I told you about a reaction is that it was non-spontaneous, what is everything you could tell me about it? The only thing you have is non-spontaneous. So tell me everything else you know. It's anabolic, right? What else? It's endergonic. Good. Is anabolic, endergonic a positive delta G? Because remember, it's an increasing, it's a mountain graph. This is where you are climbing the stairs. So it's got a positive delta G. The reactants are lower than the products. Exactly right. And it builds molecules. That is beautiful. Okay, so what's an example? Like, what's the process that is anabolic? It builds molecules. Dehydration synthesis. Exactly right. Good job, y'all. Okay, so who can tell me what feedback inhibition is? Prevents overflow. Exactly right. Prevents waste. Those are all very, very good answers. So what shuts down the pathway itself? Like what flicks the light switch? What turns it off?
The end product?
The end product. Exactly right. Exactly right, y'all. Okay, so if the only thing I told you about a reaction was that, let's make this one hard, was that an example was hydrolysis, was everything else you could tell me? Exergonic, right? Catabolic, good. What else? Negative delta G, right? Spontaneous, that's another thing I was looking for. Exactly right. What's the graph look like? Exactly right. It looks like a slide, and the product is lower than the reactant. That is amazing work, y'all. Amazing work. Okay. All right. So let me check real quick. All right. I think that is all of metabolism. Yep. Okay. So who can tell me the difference between anaerobic and aerobic pathways? That's right. The anaerobic does not require oxygen, and aerobic does. So what about the end ATP result? How do they care? Like, what's the end amount of ATP, the end amount of energy? Which one produces more?
Aerobic.
Exactly right. Exactly right. So remember that aerobic makes around 30 and anaerobic only makes 2. That's exactly right. Okay, so I want you all to tell me everything you can about glycolysis. What's your starting molecule? Glucose. Beautiful. That's exactly right. Okay, so where does glycolysis take place? Cytoplasm. Exactly right. So is it anaerobic, aerobic? What is it? It's anaerobic. Exactly. Exactly, because it doesn't use oxygen. It doesn't care if it's there or not. Exactly. Okay. What are your ending products of glycolysis? 2-pyruvate. Okay. How many ATP? 2. Exactly. And you make something else. What else do you make? 2 NADH. Exactly right. You all are giving me so much hope. I love it. Okay. So now let's move on to the prep step. Where does the prep step happen? In the mitochondria. Exactly right. So is it aerobic or anaerobic? Anaerobic. Exactly right. It doesn't use oxygen, so it doesn't care if it's there or not. Okay. So what are the end products? 2-acetyl CoA, exactly right. And you make 1 of something else. 1 NADH, exactly right. Exactly right. Okay, citric acid cycle. Where does it happen? And I forgot what I was asking. Whew. Mitochondria. It happens in the mitochondria. And it's aerobic. You beat me to it. That's exactly right. It's aerobic and it happens in the mitochondria. How many times does the cycle happen? Twice, exactly. Why? Exactly right. Exactly right. You start with 2-acetyl CoA. Okay. So, what are the end products of both cycles? So both cycles of the citric acid cycle, what are the end products? That's exactly right. So you have 2 FADH, 6 NADH, and 2 ATP. That is completely right. Good job. Good job. Okay. Now we're going to move on to the electron transport chain. Where does it happen, and is it aerobic or anaerobic? Mitochondria, and it's aerobic. Exactly right. Okay, so what happens in this step? What is set up? Proton gradient. And you're exactly right, Alexa. It doesn't produce anything. It just sets up the proton gradient. That was about to be my next question. What does it produce? It doesn't produce anything. It sets up the proton gradient. That's exactly right. Okay, so now let's move on to chemiosmosis, which I know we haven't talked about in lecture yet. So let's just get through as much of it as we can. Is it anaerobic or aerobic? And mitochondria or cytoplasm? Exactly right. It is aerobic and happens in the mitochondria. Exactly right. So for chemiosmosis, I'm not going to ask y'all about the mechanism yet because I think you're going to have to listen to her explain it and draw pictures and go in depth to really understand it. But who can tell me the end process? It's late, y'all. My tongue is tripping over itself. What are the end products? See, you got it, Carly. Thank you. 30 ATP. That's exactly right. Okay. So that is all for cellular respiration. Do you all want me to ask you questions about mitosis? No. Okay. Do I have a differing opinion or a second? All good. Okay. Not really. I totally understand. We haven't gone over it yet. Okay. All right, guys. So I already took my photo, so I will let the rest of you go. If you have any questions, feel free to hang back. If you want me to review mitosis with you, you can stay. I'm totally down for that. But I'm not going to hold you guys here. If you don't have any questions, if you're good to go, feel free to email me. Please start your 5-day study plan tonight. Please, I cannot stress that enough. So have a good night, y'all, and email me with any questions.
Jasmine, for the SWOT thing,